Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about photosynthesis. I love photosynthesis because it gives me two things that I need. I need to breathe, so it gives me oxygen, and I need to eat, and so it's going to give me food. And so I love photosynthesis. You might think it's only found in these things, plants, but it's also found in bacteria, it's found in algae, and so it's found in protists, it's found everywhere. And so photosynthesis has been around a long time. It's super important that you understand how it works. And so let's start with the site in eukaryotic cells of photosynthesis, and that's the chloroplast. So this is a number of cells, and you can see how many chloroplasts we could have in a typical cell. So there's a whole bunch of them. There are a few terms that you should be familiar with and where they are. First one is a thylakoid membrane. Thylakoid membrane is going to be organized like this. And basically, that's where the light reaction is going to take place. If you got a stack of thylakoids like this, together we call that a grana. The other big thing to understand photosynthesis is that this is filled with a liquid, and that liquid is called the stroma. That's going to be the site of the Calvin cycle. If we were to grind up a leaf, what we would find is that there's not only one pigment, chlorophyll A, that does photosynthesis, but there's a number of them that are working together. And so if you grind up a leaf into some chromatography paper, and then you put it in a solvent, what you'll get is chromatography. It's going to separate into all its different parts. And so this right here would be chlorophyll A and chlorophyll B, and this would be like carotene and xanthophylls, and they're all working together. You'll see these other pigments in the fall when the chlorophyll moves back into the leaf and is reabsorbed. But if we look at what light they absorb, here's chlorophyll A and here's B. This is what's called their absorption spectrum, what color of light they're able to absorb. And you can see that they absorb a lot of the blue, a lot of the red, but they don't absorb a lot of this in the middle, this green. And so a quick question could be, what is their least favorite color, plants? And the right answer would be green, because they reflect that green light. Now, this has actually puzzled scientists for a long time, and we really don't have a definitive answer as to why plants are green. Know this, that if they were black, they probably would get a little bit too hot. They would absorb too much light. And so let's start with an equation, because this is simply a chemical reaction. It's a chemical reaction with a number of steps. But what are the reactants? Water and carbon dioxide. And so how does a plant grow? It's basically taking water in from its roots, and it's taking carbon dioxide in through its leaves, through its stomata. The other thing it needs is light. And so it's just taking these simple ingredients, and then it's weaving those together into glucose, this monster molecule here, and then oxygen. And so this is the food that I get, and this is the oxygen that I breathe. Now, are plants just nice? No, they're making this sugar for themselves, so they can break it down using cellular respiration. And in fact, if I put this arrow in the other direction, that becomes cellular respiration. So they're making food for themselves, and they're also going to make some of the structures. So like the cellulose in the cell walls of a plant is made from that as well. Okay, so whenever I try to think what are the different steps in photosynthesis, I always imagine this picture right here. There's photo and synthesis in the word. Photo means light, and synthesis means to make. And so there are two steps in photosynthesis. The light reaction, and those are going to take place in the thylakoid membrane, and then the Calvin cycle. We used to call this the dark reactions, which is a silly term. It doesn't happen during the dark. It happens during the light. And so basically, the person who worked this all out is Melvin Calvin, and so we named it after him. Whereas this takes place, you guessed it, takes place in the stroma, or this liquid portion. And so let's kind of do a cartoon version of photosynthesis. What are the reactants again? Water, light, and carbon dioxide. What are going to be the products that come out of this? It's going to be oxygen and glucose. So let's watch what happens. In the light-dependent reaction, water and light go into the thylakoid membrane, and they produce two things. They produce oxygen. Oxygen is simply a waste product. And then they're going to produce these chemicals, NADPH and ATP. So they have energy now. Let's watch what happens to them. Well, the energy is going to transfer to the Calvin cycle, where carbon dioxide comes in and then glucose goes out. And so this is the big picture of photosynthesis. But now let's kind of dig in a little bit deeper and talk about the light reaction. Okay, so where are we? We're in the thylakoid membrane, so we're in this membrane right here. So if we were to zoom in to that membrane right here, that's what this diagram is. Okay, so what are the two things coming in? Well, the first one's going to be light. So light's coming in here, light's coming in here. What's the next thing that we're going to have coming in? And that's going to be water. Okay, so let's look at some of the other big features in this thylakoid membrane. So this is the outside, or the stroma, and this is going to be the lumen, or the inside. And so there's a couple of big things right here. What's in here? Well, these are basically going to be proteins with chlorophyll on the inside of it. And so we call that whole thing together a photosystem. So this first one is actually called photosystem 2, and then we go to photosystem 1. And the reason we go backwards is that photosystem 1 was discovered first. So basically, what comes in? Light. What's that light used to do? Well, that light is used to power the movement of an electron through an electron transport chain. So that electron is going through proteins, carrier proteins, and eventually that electron is going to go to here. It's going to go to NADPH. Because remember, that's one of the products of the light-dependent reaction. Okay, what happens to the water then? So the water is going to be split right away. If you split water, what do you get? Well, you get oxygen. So that's the O2 that's going to diffuse out of a cell, and that's the oxygen that you're actually breathing right now. And then we're going to have these protons, which are simply hydrogen ions. So they're hydrogen atoms that have lost their electron. Okay, so this is getting kind of messy. So let's look what happens next. As that electron moves through the electron transport chain, and again, it's powered by the introduction of light here and light here, that electron is going to be moving all the way down here, and every time it goes through one of these proteins, it's pumping protons to the inside. So it's pumping protons to the inside. Now, protons have a positive charge. So basically what's happening is that you're building up a positive charge on the inside. So there's a positive charge in here. If you know how cellular respiration works, you'll realize that this is the opposite of that. So now we have all these positive charges on the inside. Where do they go? Well, there's only one hole that they can go through, and that is to go through this protein here. As those protons move out, they're moving through a protein called ATP synthase, and it works almost like a little rotor. And every time a proton goes through, we make another ATP. So what have we made in the light-dependent reaction? We've made NADPH and we've made ATP. And what's nice about that is they're now just sitting right here in the stroma, and so they're able to go on to the Calvin cycle, which is going to be the next step in this process. And so who's providing the energy? Light. Who's providing the electrons? Water. And then a base or a waste product of that is simply going to be oxygen. Okay, let's go to the Calvin cycle then. So what's happening in the Calvin cycle? You can see here's those reactants. So we've got our ATP here, and NADPH. What are they providing? Simply energy. We also have this molecule here. It's called RUBP. Basically, it's a 5-carbon molecule. And then we have carbon dioxide coming in. So it moves through the stomata of the leaf and it's going to diffuse its way in. Carbon dioxide is a 1-carbon molecule. So basically there's an enzyme here called Rubisco, and it's going to attach this 1-carbon molecule to a 5-carbon molecule. It immediately breaks into 2 3-carbon molecules, and then it gets energy from ATP and NADPH. And when we're done, it's creating this chemical down here called G3P. What does G3P become? Well, it can be assembled quickly into glucose or sucrose or maltose or whatever they need to do. That's going to be produced right in here by the G3P. So that's where we're synthesizing. In other words, we're taking carbon and we're fixing it. We're making it usable. Now, some of that G3P is released, but a lot of it is recycled again to make more of this RUBP. And so that's why it's a cycle over and over again. What's the big picture? If we don't have ATP, if we don't have NADPH, then this process is going to shut down. What's the other thing that could shut it down if we don't have carbon dioxide? Okay, so that's basically photosynthesis. And again, it's been working for billions of years. But there's a slight problem, and that problem is called photorespiration. What is photorespiration? Well, photorespiration occurs only when we don't have enough carbon dioxide. So if we don't have enough carbon dioxide, let me cross that out. Well, we certainly can't make our G3P, but something worse happens. Oxygen can actually jump into the Calvin cycle and using Rubisco can form another chemical. Now that chemical doesn't do anything. In other words, it has no purpose and the cell actually has to break it down. And so as a result of that, plants, and we call almost all plants C3 plants, and the reason we call them C3 plants is this G3P is going to be a 3-carbon molecule. So these C3 plants, photorespiration is bad. In other words, they don't get anything out of it. And so they're going to lose based on that oxygen kind of jumping into the Calvin cycle. And so you might think evolutionarily, why would this have even evolved? Well, remember, photosynthesis shows up first and then oxygen in the atmosphere shows up much later. And so it wasn't a problem initially, but it became a problem. Another question you might think is, well, when are we not going to have enough carbon dioxide? When wouldn't we have carbon dioxide? Well, how do they get carbon dioxide? A plant is going to have a stomata, and it's surrounded by guard cells. And so basically, when a plant opens up its stomata, carbon dioxide can diffuse in. And so the only time the plant wouldn't have carbon dioxide, because we have tons of carbon dioxide in the atmosphere, is when it's actually closed. And when would it be closed in a plant? The only time it's closed is when it's really, really hot, and a plant doesn't want to lose water, because through transpiration, you're constantly losing water. And so if you're a plant, you have this, if it's a hot day, you have this really tough choice. If you open up your stomata, you're going to lose water, you could shrivel up. If you close it, you can't get carbon dioxide in, and then you're going to start doing photorespiration. And so, of course, nature has come up with solutions to this over time. And it's only going to be found in plants that live in a really hot environment. So here's the first solution. And this totally makes sense. So this is in camp plants. Camp plants, an example would be a jade plant or like a pineapple. Basically what they do is they only open their stomata at night. And so at night they open up their stomata, and then the carbon dioxide will come in and they'll create malic acid out of it. So they're going to store it in vacuoles inside the cell. Okay, so now when it's daytime, what they can do is they can close the stomata because they don't want to lose water. And now they can actually take that carbon dioxide out of the malic acid, and they can use it in the Calvin cycle to make sugars. So the great thing about a camp plant is, again, they're only taking in carbon dioxide at night when it's cool. And then during the day, they can close their stomata and they don't lose water. Another example of this would be in C4 plants. What they do is, instead of doing it day and night, what they'll do is they'll take that carbon dioxide in, and they'll actually use enzymes to make a 4-carbon molecule out of it. That 4-carbon molecule will move to some cells on the inside of the leaf called the bundle sheath cells, and then they can simply introduce carbon dioxide into the Calvin cycle here. And so again, both of these solutions are basically taking in carbon dioxide when you can get it, creating a chemical out of it, and then they can introduce that chemical into the Calvin cycle, and they don't have to wait for carbon dioxide to diffuse in. Now, of course, there's going to be extra steps in here, so it's going to require more energy, and so we only see this in areas where it's really, really warm. But an example of a C4 plant that we all eat and use a lot of, in fact, most of us are just made out of this stuff, is corn. And so that's photosynthesis. A simple problem is photorespiration, but I hope that's helpful.